because you're going to learn how to do preclinical studies, you need to understand how to do the experimental designs. And it means that as you do that, you should have an idea about what is meant by scientific rigor and how you can go forward to make sure that when you design your studies in the future, that will be implemented in your experimental design. And that's very important because that's the best way to get good translation from the preclinical arena to the clinical arena. It's also a requirement by funding institutions such as the NIH that you incorporate that into your grants. And then finally, I'm going to default to probably the most commonly used rehabilitation methods. Just to give you some other information about that, something for you to think about as you go down this path of saying, I do regenerative medicine, I want to implement some kind of exercise program. Maybe what I'm going to do is I'm going to look at running wheels or treadmills. So I'm going to tell you there's a little more to the story than that. And I'll end kind of with an experimental design to give you an idea of how complicated this can be and what to think about before you even start. The next is a historical piece about experimental rigor. I see all of you very interested in doing regenerative rehabilitation. And I also envision it as being a complex paradigm when you think about how you're going to design your experiments. So let me take a few steps back and give you some history on the importance of experimental rigor. So this came from The Economist. It was published in 2013, and big, bold letters about trouble at the lab. Trouble at labs running preclinical studies. This is not my quote. This actually came from The Economist, but I do think this way, that I think that if I do a study in my lab, I should be able to replicate that same study in my lab, and someone else in a different lab should be able to replicate it too. That doesn't happen very often. People don't even try it. And we don't know the degree to which, if you do try it, whether you actually replicate your own results. But it is, this idea of replication is really considered the cornerstone of the type of science that we do. It makes for good science. And honestly, if your true commitment is to improving the human condition, then you have to be able to show that whatever you're doing, someone else can replicate, and or at a minimum, that you can replicate it too. Why did this all happen? In 2012, there was a groundbreaking paper that appeared in Nature. How many of you know about this paper? Okay, so maybe about 15% of you. So it brought together stakeholders, those involved in publication of your papers, those involved in funding your grants, etc., the pharmaceutical industry, because they thought that there's a lack of transparency in how publications are currently produced. And they're not predictive of, the preclinical research is not predictive of what happens in the clinical arena, which is obviously a problem. It's a problem for us all. So what went on in that meeting was a review of several interesting things that happened, and I'll just give you just an overview of that. The first was that in 2011, Bayer Healthcare went back in-house and tried to replicate their studies. And to their surprise, out of 67 preclinical studies in this system, they could only replicate 25%. Doesn't that kind of worry you about what's going on in-house? And you think that what you're doing are really, vigorous studies. Amgen then took another step. They tried to replicate 53 studies that they called their landmarks, in-house landmark studies. And this happened to be in cancer research. And the company was only able to replicate 6 of those cases, about 11%. What I won't tell you here, but if you go back and you look at the history of this, we're not talking about when you publish publishing in low-impact journals. This problem, they showed, was across all journals, independent of their impact factor. So finally, and I don't know if this was a turning point for NIH or the fact that the preclinical models were not predicting well for the human condition. NIH had a contract to a group of centers to replicate published studies on drugs that would be efficacious in spinal cord injury. These were already published studies. So they attempted to do that, and very few could actually, you know, in a well-controlled, communicating with the previous authors of those papers, very few could actually replicate. And I would even challenge the one that said that they could replicate. And it was in part, I mean, there may be many reasons for this, but it was in part due to the fact that you couldn't replicate it if you wanted to just by virtue of how they described what they did in the paper, which is this issue about lack of transparency. If you want your colleagues to help you validate what you've got, you have to make sure that you've written it very clearly. They were incomplete or inaccurate, and when it came to the how they designed the studies, it was difficult to know if they randomized, and if so, how they randomized, how they formulated their groups to study efficacy, and even reporting of the total number of animals that you started out with. How many did you lose along the way, and why did you lose them? What was that? What was the cause of that attrition rate? And these were all problems that existed in these papers. So as we talk about regenerative rehabilitation, we're talking about not just one means to improve outcome, we're talking about combining two. So it becomes a much more complicated experimental paradigm where you have to be very careful about how you think about this. An exciting area, but one that be aware. So then in 2016, Nature goes back and asks about, they surveyed about 1,600 researchers. And they asked him, do you think there's a problem in reproducibility? Now remember, this is about 5 years later. So 52% said, yes, we think there is a problem with reproducibility. But there's still about 38% who say, if there is, it's going to be really minor. So the reason why I'm emphasizing this with you today is I just don't want you to be the other 38%. Because it is a big deal. And I think we're all committed to improving the human condition. And so if what that takes is a really rigorous and transparent design, then that's what we should be doing. That's what we should be moving forward down this pipeline to improve outcome. If in the same review, they asked, what factors do you think contribute to irreproducible research? So I don't know if you can see it because it's pretty light, but the top one was selective reporting, meaning that, okay, you report the things that really had great outcomes, but you don't report the ones that maybe had negative outcomes. And one could argue, well, it's very hard to publish negative findings. Actually, there are journals now that will embrace that concept. Another was, just want to go through the top three, the pressure to publish that we all feel vulnerable to, but we should not succumb to that until you feel that what you have are really rigorously designed studies that you have executed to your satisfaction. And then one that I'll put myself into this bucket. When it comes to statistical design, to power power analysis and how we analyze our data, most often my experience has been in the preclinical literature, it's pretty weak. And I put myself in that too. We weren't trained, my generation wasn't really trained on how to do a preclinical trial properly. So part of this is, if I say to all of you before you start your study, first do a power calculation to figure out how many animals you're going to need, how would you what would you do to determine that number? So I'm putting it out there because these are all things that you need to do before you even start a study that involves regenerative rehabilitation, or any study actually, where you hope to translate to the clinical arena. So the crisis in numbers, how much published work in your field is reproducible across multiple disciplines? Well, the people that were most confident about this were the chemists, the physicists and engineers. The rest, not so confident. It's easy to get freaked out if you start calculating numbers in your mind, and I'll show you why it'd be easy to get freaked out when you think about it. A lot of times when you're thinking about preclinical trials and you have ballpark numbers for looking at behaviors, say you're looking at neurological recovery. Oh, that's about 15 animals per group if you're just doing it in your head. Now double that when you're doing both males and females, and you start thinking, and then a treatment paradigm, or in the case of regenerative rehabilitation, you do one treatment, each one separately, and then combine, right? So this is where your really good statistician can help you in designing these studies so that you don't necessarily have to do it the way you've always thought. And I would encourage you, as you go down this path of planning these studies, to bring a statistician on board. Because there are ways to deal with how you ask your questions so that it's not necessarily the numbers that you think they're going to be for that particular study. So just in summary then, when you're designing a study, consider the scientific rigor, consider sex as a biological variable, and use your colleagues, experienced colleagues. Your local statistician will be a godsend. And NIH has terrific tools for learning about all of this. Not only to help you design your experiments, to help you get funded, but these sort of rules are actually flowing into other funding streams as well. So it's important that you really take this on and design these properly. Okay, so I think I've preached enough about scientific rigor. So now I'm going to tell you about two common tools that are used in the trade for rehabilitation. Two of the most common, especially in the neurosciences, which is where I come from. So we'll talk about, just briefly, voluntary and involuntary exercise in rodents, the use of voluntary running wheels, and then treadmills. So I just wanted to share a few observations about this. So what are the strengths of a running wheel? Because people oftentimes debate what they should be using. So if you happen to have a high-end computerized system, it will give you feedback on the diurnal activity of those animals, because they are very diurnal in their overall activity. You can determine their running speeds, and their bouts of running can be tracked. You get all this information if it's computerized. The limitations can be if what you're using are not it's not a computerized readout, but it's like the clickers, where the clickers determine how many times the wheel's moved, or odometers. They're really unable to capture the kinds of information that would really make your study better. That is, the speeds, the running speeds, these bouts of running, and diurnal activity. So you have to weigh in on what you have and what the value is of a running wheel. So now we were talking about sex as a biological variable. I just wanted to show you, this is running wheel data looking at males and females, and I think this was in the C57 Black 6 strain. So these are days of exercise in both cases, and you're seeing time spent running and distance run. And what stands out here is the degree to which the females, the patterns of female running, which is quite different, longer distances, more time spent running than their male counterparts. How many of you knew that? Okay, so I thought that was interesting. And then if you have this computerized system, this just shows the days of exercise. So, and you put them all together, and you can see the group sizes here, up through about day 14, there's a significant difference between the females and the males. With the females, the average speed is higher, and then they kind of flatten out, and they're similar to your males. So if you're trying to do the running wheel, you ought to know these kinds of things exist. And that you have to be cognizant of the fact that rodents are diurnal. And so if you have the ideal scenario, which is one of these computerized systems, you can track how often, what these animals are doing during periods of light and periods of darkness. Now, if you look here, these are males and females. This is a period of darkness. And again, you can see the distinction between the females during this period of darkness compared to the males in terms of the distance run. They do a lot more than the males. What about treadmills? We won't say too much about this, but they are one of the key strengths of this is that you can have these animals run a defined distance at a defined speed. You can change the angle of the treadmill, so you have all these variables you can do, and you probably come out with very clean numbers. The limitation of these is they're often criticized because in some cases it's non-physiologic, what you're asking them to do, and good researchers won't make this mistake. They can be stressful. I mean, stressful in the biologic sense, too. If you read the literature, you will come across some where they actually do this with a black box over it so it's in the dark. But the reality is most of these are done in daylight hours, which kind of, for an investigator, that's how you do it. But the fact is that they're most active at night than they are in the daytime. So if you go down this path, you have to realize that maybe what you need to do is design a system where you can do this in a darkened room. So this is treadmill exercise. I want to introduce another aspect to think about when you're trying to add this to your preclinical regimen. There are strain differences just in mice alone on how they perform on the treadmill. This is monitoring a beam break. So as the rodent is running on the treadmill, that's going this direction, they start getting tired, or for whatever reason they start slowing down, and the back end of them hits a beam which breaks, and you can calculate how many have had a problem maintaining their position on that treadmill as it's moving forward. That's also the place where there's a mild shock that they give to promote them to keep running on the treadmill. So there's differences in the beam breaks that you see here, as well as the speed itself. So when you're doing these kinds of studies, think about the kind of mouse that you want to work with. The one that stands out here in terms of just the number of beam breaks is the C57 Black 6. The one that's not keeping up as well as some of the other counterparts. The trade-off on this is a lot of people study the C57 Black 6, so sometimes it's hard to compare with other groups if you turn to some of the others, but you should be aware that that's kind of a feature of this strain of mouse. In voluntary wheels, there's also strain differences in running duration, in running, and running duration, running distance, and average speed. So again, do your homework on the voluntary wheels as well, and trying to pick the strain that you want to work with. That will answer your question the best. So, so let's put them together, and I'll tell you something really random, which is if you put mice on a rotor rod, they just naturally go round and round, and it's not really a very stressful situation for them. So there was a student in the lab who wanted to learn how to do the rotor rod. So we looked at 2 strains and 2 sexes on this, just on the rotor rod. And so what you see here, move them all down. What you see here is if you don't separate them out by sex, the CD1 and the C57 Black 6 don't show any difference on the latency to fall on the rotor rod. They look about the same. If you break the data down and look at the CD1 males and females and the C57 Black 6 male and female, what you find is that you reach a difference in one group, this group, this group, the CD1s, in terms of latency to fall. So, which sort of speaks to the fact that it's a combined, I think, reminder of thinking both about strain and sex of the animal that may vary even on an activity like the rotaride. And finally, when you're doing exercise of any kind or any manipulation of the animals, just bear in mind that that's impacting them in other ways in terms of how they feel about their environment. I just put this in here. This is an open field, and you're just tracking where the animal goes in an open field. Rodents, people too, prefer perimeters. They feel a lot safer there. They don't really, in general, spend much time in the center of it. And if you look at total distance moved between males and females in each of these strains, there's no difference there. Or in their total rest time, there's no difference. But if you look closely at the time spent in the center with the C57 black 6, there is the males tend to spend more time in the center than the females. It's not an abnormality. It just means that there's differences in the degree to which they are comfortable being in the open field compared to the perimeter. It's just, I think, a reminder to us all that as you do these studies, bear in mind that there are aspects of how you handle these animals, et cetera, in preparing them for their treatments that can affect how they perform. Then I think we're close to the end. This concept of phenotypic stability, if you're using running wheels, if you put a mouse in a running wheel, and it's when you start your study, what you'll find is a high degree of variability, because what you have to do is get them used to being in a running wheel and defining their standard time that they spend in the running wheel. And the degree to which they need that will vary depending upon what you're looking at. So running distance, duration, and speed can all require a different amount of time to get them used to that. So if you're designing a study and you've elected to work with running whales, bear in mind that they need this period of time to get used to it. You get readouts to make sure that the true test is when they reach a stable performance period. That's when you know you can stop. And it can be anywhere from 3 days to 7 days to make all these parameters stabilized that I mentioned here. Right? All right, so I thought I would just end with this. This is like the old-fashioned way of designing a study where you're not even going to have a treatment. You're just going to look at males and females. And we're going to have the running wheels embedded in this. So every number that I put in here, think about this as we're not even introducing a rehabilitation method here. So that would increase the animal numbers, right? This is why the standard method that I'm describing here is probably not what you should do, but this is the kind of the traditional way of how people have done it in the past. So you're now fully knowledgeable about the strain behaviors, and they match well with the question you're going to ask. You know the age of the animal that you're going to work with. You have done the a priori power calculations that tell you the group sizes. You've specified your inclusion and exclusion criteria. You're going to randomize. You can indicate how you're doing the randomization, and there are computer programs for that. And you defined your endpoint or endpoints. This is all done. So you need a 7 to 14 day acclimation after they've arrived. Oh, and you've determined that you're going to have 75 males and 75 females. They have they come from the vendor. You need to let them acclimate to their new environment. And somewhere between 3 and 7 day exposure to the running wheels, so they all perform in a regular way. Now, there are 25 males and females who do not meet your a priori criteria for how they're performing on those running wheels. They're not stable, even after up to 7 days. So you're going to exclude those, and that's part of your exclusion criteria. So now you're down to, these are round numbers, you're down to about, you're going to randomize 50 males and females, some that will have exercise, no exercise. You just put the wheel in the cage and you lock it. So they can go in and out of it. They have that physical structure in there, but they can't do any exercise. Then you have one that has the actual running wheels, and you can map what's going on. And as you're running all these animals, now you've got 25 in each group, you're going to have some exclusions. Some things just happen, like, for example, the failure of a running wheel, or something happened to some animal. So you end up with, you know, at the end, you're going to end up with fewer animals than what you started with. We've all had that, where we begin large and then we have to cull out as we go along, and then you analyze your data. So those are just some of the basic principles of how you think about what you have to do beforehand before you start this study. And the reason why I give this to you, and I know the numbers are large, because I think my ballpark figure on doing behavior is like about 15 to 20 animals in each group. That means you completed 15 to 20, so you start larger. But that's without doing a power calculation. Now, 2 treatments, stem cells, let's say, and a rehabilitation one, and think about how that would change the numbers. Think about why it's important to have a statistician who will help you in the design of these experiments based upon the types of questions you're going to ask. And it's all doable, but having that partner alongside of you will make your life a whole lot easier, and you'll end up with a much better study. I'm making the assumption that you don't have a good statistical background, as I have. So it's been a lifesaver for me. All right. That's all I have to tell you that I know. I've tried to touch upon the things that would help you succeed in the future if you haven't done this before. I hope it will compel you to go to the literature and review the kinds of rehab you can do. It goes far beyond running wheels and treadmills, electrical stimulation, ultrasound, all interesting areas, hot and cold. And then think about how to make the design of those studies highly vigorous, taking into account that you have all this arms to your study.